to study morphology, microscopy and powder characteristics of ephedra. Synonyms: ephedra is also called as mahuang. Biological source of ephedra: dried young stem of Ephedra gerardiana staph and also of Enebrodensis staph collected in autumn. Family: Genetaceae, Ephedraceae. It contains not less than 1% of total alkaloids, calculated as ephedrine. Chemical constituents: alkaloids 0.5 to 2%. Alkaloidal amines: ephedrine 30 to 90% of alkaloids. Uses: it is sympathomimetic. It causes mydriasis in asthmatic condition.
To study the microscopical characteristics of ephedra, we need the following experimental apparatus. Cutting of fine and thin section. To study the microscopical characteristics of a crude drug, it is essential to have a thin section of drug. A fresh blade is used precisely to cut the section of ephedra as seen in the video. Staining process. Stain is used to make cells and other components more visible and to highlight specific features. It is prepared by mixing fluoroglycinol and HCl in the ratio of 4:1 on a watch glass. Staining of section. The section of ephedra which needs to be stained is picked carefully using a fine paint brush. Immersed in the HCl and fluoroglycinol mixture previously prepared on a watch glass. Preparation of slide. 10 minutes after stabilizing the section, it is kept on the slide along with 1 drop of glycerin and adequate amount of water droplets. Adding excess of glycerin makes cover slip difficult to handle and results in poor quality of images under microscope. With the help of pharmacognosy needle, place the cover slip over the slide. Make sure that no bubble gets entrapped during this step. Observation of slide. Carefully place the slide under the microscope and observe the section under 10x lens. As seen in the video, ephedra has a unique microscopical structure which includes components like epidermis, cortex, lignified and unlignified fibers, pericyclic fibers, xylem and phloem and pith. Powder microscopy. At first the powder drug is added to the fluoroglycinol and HCl mixture. Adding drug to this mixture enhances visibility of certain cellular components and structure. After a few minutes, a smear is carefully prepared on a slide using a fine paintbrush. After placing the cover slip, the section is observed under the microscope. Through this technique, histological analysis and quality control of drug can be evaluated. Ephedra powder has distinctive microscopical structure comprising epidermis, fiber, xylem and brownish matter. Epidermis consists of quadrangular cells and its outer walls are ridged. Fibers are lignified and non-lignified, long and cylindrical. Xylem appears as tracheids with bordered piths. Brownish matter are abundant, dark brownish mucilaginous substance from pith.